Well, hello again. It's Kevin Kyler from Environmental Inspection Dynamics. The two preceding videos might have brought you down a little bit, so I'm thinking about bringing you back up a bit. And with this question, is there a good side to mold and fungi? So let's go on. And the answer, yes. So let's take a look. Let's see. Present sciences and studies are showing us a lot of new discoveries occurring that were hidden. So we're going to go over 5. The what happened at Chernobyl, the what might be used on trips to Mars, the what that's happening from King Tut's curse, the what that's happening with reforestation, and the what that's happening in cancer immunotherapy. The what at Chernobyl. Remember that? There's some pics. Take a look at that. Looks like mold to me. Ooh, that scare you? What is this stuff? The mold species that was found at Chernobyl is known as Cladosporium sphaerospermum. What a name, huh? So, here's what the scientists found out about Cladosporium sphaerospermum in the ruins at Chernobyl. It thrives under radiation. The melanin in its spores absorbs the deadly radiation and converts it to energy by a process called radiosynthesis phenomena. That's a tongue twister. The what that might be used on a trip to Mars or even the space station. What? Back to Chernobyl? From Chernobyl to Mars. Now let me ask you a question. Do you think there's no radiation on the trip to Mars and on Mars? So I'm going to read this. Now, the melanin in the spores and other species of mold can absorb the deadly radiation and it turns to black mold. This is from the National Institute of Health's National Library of Medicine. And I read, the catastrophic Chernobyl explosion on the 26th April 1986 released approximately 108 curies of radionuclides into the environment, rendering the area uninhabitable to this day. Now, scientists are trying to see if this mold pigmentation would protect astronauts from dangerous radiation. What do you think about that? Now, let's get back down to earth here and look at the what's happening from King Tut's cursed tomb. First, an introduction of a few guys. Howard Carter and Lord Carnarvon. All right, I'm going to read this. The supposed curse that killed the archaeologist, which included Carnarvon, was Aspergillus flavus, which causes severe respiratory infections. Aspergillus flavus spores can lay dormant for centuries. This same fungus that killed shows promise as a treatment for what? Leukemia? All right. Derived from Aspergillus flavus are Aspergillomycins. You pronounce it whatever way you want. That's the closest I can get. The same research team that discovered this also believes that similar life-saving compounds may come from other fungal species. What do you think about that? Now look, you think I'm making this stuff up? I'm not. There's proof. Now here, get your shovels. We're going to dig down a bit with what's happening with reforestation. All right, this will be the antithesis of, you'll get it, root cause analysis. This is about fixing global forest degradation because of what? Climate change. Pay attention to what I underline. It'll come together. This is done through plant mycorrhizal synergy. We're going to focus right there in those zones, down where the roots are, just above the surface. Now, this was led by the South China Botanical Garden of Chinese Academy of Sciences in collaboration with Germany, us, Czech Republic, Netherlands, and Italy. Done through, here we go, a bruscular mycorrhizal fungi. What? Which acts as deep carbon engineers. Another one, also ectomycorrhizal fungi, functions as surface active stewards. You getting below the surface and above the surface. This mycorrhizal inoculation with native, efficient fungi helps rebuild soil microbiomes, improving plant survival and stress tolerance. This plant mycorrhizal synergy showed 40% higher soil carbon storage. Now look at the underline. We got climate change. Deep carbon engineers get 40% higher soil carbon storage? Again, I'm not making this up. But what's happening in cancer immunotherapy? Now, if y'all want, you can stop this video and quit listening to me yik-yak at you and study that. However, I got something else to read. A little longer, but sit back or follow me, whatever the case. The details. The researchers developed a method to feed T cells glucose without offering it to the tumor. They turned to cellulobiase, a naturally occurring sugar found in plant fiber that human cells and tumors cannot break down. Look at this. But some microbes and fungi can. By equipping T cells with two proteins derived from fungi. The researchers enabled the immune cells to import cellulobiase and convert it into usable glucose inside the cell. In lab experiments and the mouse models, the engineered T cells stayed alive, continued dividing, producing cancer-fighting cytokines, and effectively killed tumor cells, while the unmodified T cells stuff rapidly lost function. This study was published in the Journal of Cell on, look at the date, February 25, 26. I'm going to read you a little bit more into this. It's called What the CAR T Therapy Is. Follow me along. The CAR T, or chimeric antigen receptor T cell therapy, is a form of cellular immunotherapy. It's used primarily to treat certain blood cancers, such as B-cell acute lymphoblastic, leukemia? You mean mold is going to help us fight leukemia? You guessed it right. You want more information? Then go online and let AAI help you find it. The main purpose of this? Look, we need to live with mold and we need to work with it. You get and understand it's not all that bad. All right. Now, I know some of you pundits in this area have more knowledge than I have if you looked at this. Glad to talk to you. If I need correction, so be it. Well, listen, folks, I hope you enjoyed the lighter side of the good side of what mold can do. Thanks for watching.